Hello guys and welcome back to Excel Share Academy. Now as part of cell biology CSS syllabus, we will be moving to our next topic, and that is cell structure and function, of which nucleus will be first we will be discussing. Okay, so when I say nucleus, everything we are going to discuss, like how protein sorting is done, like proteins are, we know that the proteins are getting synthesized in the cytoplasm, right? So these proteins are then how it is recruited back to the nucleus से कैसे आपके mRNAs RNAs आपके cytoplasm में जाते हैं, so those will be covered. How the chromatin or the chromosomes are arranged inside the nucleus. Does it have its own territory, yani ki like we have got the 23 pairs of chromosome, so is it present in the nucleus randomly, or it has got its specific location? That we are going to discuss. So everything related to how the chromosomes are being aligned, how they are being functional, right? So all this, whichever is related to the nucleus, we will be completing in this particular session. Okay. Likewise, each organelle of the cell will be discussed. Like in future, we are going to discuss mitochondria, chloroplast, peroxisomes, Golgi apparatus, etc. in detail. Okay, so this is how we are going to cover all the organelles of your cell structure. Okay, and then comes the cell signaling, the cancer, etc., etc. Okay, so let's start with first our nucleus. So the presence of nucleus, it is a principal feature which will distinguish between the eukaryotic and the prokaryotes. By housing the cell's genome, nucleus serves both as the repository of genetic information as well as the control center. So this we know, right? Depending on the environment stimulus or the external stimulus, whatever the signal is perceived by the cell, accordingly the genes are going to get expressed. Okay, DNA replication, transcription, RNA processing all take place within the nucleus, with only the final stage of gene expression, that is the translation, which happens in the cytoplasm. So now by separating the genome from the cytoplasm, the nuclear envelope, it allows gene expression to be regulated by mechanisms that are unique to eukaryotes. Now, what do we mean here? So we understand that we have got the eukaryotic cell, we have got the nucleus over here, right? So nucleus means nuclear envelope is there, which is separating from, which is separating the nucleoplasm from the cytoplasm, right? Now this barrier is helping in regulation. It allows gene expression to be regulated by mechanisms. So what are those mechanisms? So these mechanisms are nothing but, for example, if you remember in the previous lecture, we have talked about NF kappa B. So NF kappa B is what? Your transcription factor, correct? If you remember. So these transcription factor are what? They are in nature, by chemistry, they are protein. But where it is required? It is required in the nucleus, correct? So when you get a signal, then only the NF kappa B, which is masked by the activity of I kappa B, okay? So when there is a signal, this gets phosphorylated. What happens? I kappa B loses its conformational change and NF kappa B becomes free. When NF kappa B is free, then only it enters the nucleus, right? So what is the mechanism? This is the regulation we are talking about. The gene expression to be regulated. So this is the regulation, that you don't allow all the transcription factor present in the cytoplasm to go inside the nucleus. Whenever there is a particular cell signal or stimulus is given, then only these transcription factors will go inside the nucleus for that particular gene expression. Is this point clear to everyone? Yes? Akshaya?
Yes.
Yeah, okay. So whereas in the case of the prokaryotes, mRNAs are translated while the transcription is still in progress. Now, this is like, you know, the benefit of not having a separate nucleus. So in the case of the bacteria, as the transcription is going on, if you remember, the translation is also happening in this case, in the case of the bacteria, and this process where you have got transcription and translation happening together, it is known as co-translation. ओके तो जैसे जैसे आपका mRNA बनता जा रहा है वैसे ही आपका प्रोटीन भी सिंथेसाइज होता जा रहा है. So the presence of a nucleus allows gene expression to be regulated by post-transcriptional mechanisms such as alternative splicing. So if you remember, we have talked about the different types of splicing. Jahan pe introns are there. Now, this is the difference. Like why there is no nucleus or there is a nucleus present in the eukaryotes. See, in the case of the prokaryotes, you don't need splicing. Why? Because their DNA does not have introns. Correct? So there is no intron. So there is no requirement of post-transcription mechanism. There is no requirement of any splicing to be done. Okay. Whereas in the case of eukaryotes, we know that so much of junk DNA is present or the introns are present. So these introns needs to be spliced out before your complete matured mRNA is synthesized, before it is transported to the cytoplasm. So for that, we need splicing to be done. Agree? Yeah. Now, by limiting the access of selected proteins to the genetic material, the nuclear envelope also provides novel opportunities for the control of gene expression at the level of transcription. So what it is saying is that before reaching the stage of the translation, there are many ways by which at the transcription level itself, you can regulate the gene expression. Agar aapko malum ho, so when we say gene expression, okay, gene expression does not always mean having the proteins synthesized. RNA bhi agar synthesize hua hai, so even that is also known as gene expression, okay, and a protein of course. No gene expression means no RNAs are synthesized, no proteins are synthesized. Okay, I hope this is clear. Yeah. For example, the expression of some eukaryotic gene is controlled by the regulated transport of transcription factors from the cytoplasm to the nucleus, a form of transcriptional regulation unavailable to the prokaryotes. The separation of the genome from the site of the mRNA translation thus play a central role in the eukaryotic gene expression. So here again, the same example I can talk about where I have mentioned about the NF kappa B, right? So NF kappa B is bound with the I kappa B. If it gets phosphorylated, this goes for the degradation. This is the same figure I'm trying to make. And then NF kappa B goes as the transcription factors binds to the gene, and then you have got the gene expression. Okay, so this is not possible in the case of the prokaryotes. Only in the case of eukaryotes it is happening. Am I going fast or are you able to catch? Shruti, Tanvi? Am I going fast or the speed is okay?
No, ma'am, you are okay.
Okay.
It is okay.
Now the nuclear envelope, the traffic between the nucleus and the cytoplasm. So from here onwards, please start giving more concentration. Okay, so the nuclear envelope separates the content of the nucleus from the cytoplasm, and this is done with the help, like if suppose there are certain things which is required. This is suppose your cell, this is your nucleus. Now there are certain proteins or the other components which is required in the nucleus, okay, or from the nucleus it should go to the cytoplasm. Okay, like how we have discussed in the case of the plasma membrane that we have got the channels, right, or we have got the carrier proteins which helps in the transport of the materials. Similarly, in the case of the nucleus, we have got NPC. Now, what is NPC? Any idea? Have you heard of it? Akshaya, would you like to say what is NPC?
I cannot remember.
Okay, no issue. Tanvi, what about you?
Don't know, ma'am.
Okay, no issue. It is your nuclear
protein
pore complex. Okay, so this is nothing but it is a bridge between the nucleus and the cytoplasm. Okay, now we are going to discuss about the NPC. So structure of the nuclear envelope. It consists of two membranes underlying nuclear lamina and the nuclear pore complex. So wherever they have shown the marking, okay, ऐसे arrow marks दिए, वहाँ पे आपका NPC present है. Okay, the nucleus is surrounded by a system of two concentric membranes called the inner and outer membrane. So this is also very important. Why? Because see, the outer membrane of your nucleus is in continuation with your ER. Okay, and that's why it is like the composition of the ER membrane and the composition of your nucleus ka outer membrane, it is going to be similar. Okay, 1 second guys. So where was I? So I was talking that the outer membrane of the nucleus and the ER. Okay, the ER membrane composition is going to be similar. Okay, and then it has got the inner. So wherever NPCs are present, you can see that they are combined together. Suppose this is outer, this is inner. So inner में भी दो रहेंगे, outer lipid layer, inner lipid layer. And in the outer membrane, outer and inner will be there. So wherever NPCs are there, it will be like this. Okay, yeah. So the outer membrane is in continuous with the ER. So the space between the inner and the outer is directly connected with the lumen of the ER. Now, why this is important? This point is important because see, when a mRNA is synthesized in the nucleus, okay, and it is going to be getting translated. If that mRNA is meant to be a membrane protein, okay, that time it will directly go to the bound ribosomes on the ER. Are you getting it? Right? So the distance or the energy required to transfer the mRNA from the nucleus to the bound ribosomes will be less and it will be easy. Anyways, eventually we'll come to that. I have got the figures also. So, okay, this much is clear, I believe. Yeah, this is the figure. So here you can see that you have got the inner and outer. Now, wherever NPCs are present, you can see the difference over here, right? It is totally merged. आपका outer inner उसके साथ combine हो जाता है. And then eventually, wherever ER is है, the outer nuclear membrane is in continuation with your ER. Right? Are you able to make out? Yes. Yeah. So you can see that here the chromatins are there. You have got wherever ribosomes are attached, those are your rough ER. जहाँ पे नहीं है वो आपके smooth ER हो गए. Then this is your nuclear lamina. Can you see this portion? Now this is also very important. ठीक है, question इसी पे ही आते हैं. So you have got, see, ये आपका nucleus का first layer हुआ inner. ये हो गया उसका outer और इसी के ठीक नीचे you have got the nuclear lamina. Okay, so now lamina, they are nothing but made up of lamins proteins A, B, and C. They are made up of lamin proteins A, B, C. Now, what are lamins? They are nothing but they are your intermediate filaments. Now, what are intermediate filaments? Anyone? Shruti?
The cytoskeleton, part of the cytoskeleton.
Yes, absolutely correct. So they are your cytoskeleton proteins. Okay, so we'll do this also in detail, but please do remember because this is the mainly intermediate filaments which we'll be talking about. Lamins हो गया और ये हो गया आपका keratin. So by the name itself, you know where this keratin protein is present. आपके nails में, आपके hair में, right? And it is also a part of IF, intermediate filaments. So we'll come to that. Now, the outer membrane is similar to the membranes of the ER and it has got the ribosomes bound to its cytoplasmic surface. but of course it is going to differ in the protein composition, and that's how you understand कि ये part जो है आपका ER का है और ये part है जो आपका nucleus का है. Okay, so this is all about the integral membrane proteins present on the outer membrane of the nucleus and the ER. So basically ER में हम expect नहीं करेंगे the maximum integral proteins. Okay, it will be present in your outer membrane. So this is just the electron micrograph they have taken, and what they have shown is that this is the outer membrane. Here you have got the NPC, and this outer membrane is going in continuous with the ER membrane. Okay. Now how we have understood is that based on the types and the number of the integral membrane proteins present on the outer membrane of the nucleus. Okay. So there are many experiments people have carried out and all. So here your one interview question can come. Okay, that is like, what experiment will you carry out in order to differentiate that this part of the membrane is outer membrane and this part is of ER membrane? So what should be your answer? That try to find out the proteins, integral membrane proteins present on the nucleus, so you can have the antibodies against that. So wherever the antibodies are binding, which is having the fluorescent, it will go and bind to the outer membrane region, and where you don't have, it is going to be your ER. Okay, so ऐसे questions आ सकते हैं, ठीक है? And that's why for example, recently also have taken one mock interview, and there same, even though you know the concept, okay, the same figure also they show in the interview, during the interview. You have studied it, but the question will be twisted कि आप कैसे पता करोगे, ठीक है? So that's why these reference books are good, because they give you the details like how they have conducted the experiment and what to interpret and how they have concluded. So इसलिए reference book से पढ़ना अच्छा रहता है हमेशा. Okay, anyways. Now look at this figure. Okay, so this is again, you can see that many nuclear pores are visible over here. Now whenever you get such kind of images, you should get that this is your freeze fracture technique method. ठीक है? तो जहां जहां पे आपको dot आ रहे हैं. Now how they understood that this is NPC? Wherever, see, अगर आपके पास कुछ चीजें हैं और आपने इन दोनों को combine कर रहे हो, so obviously there will be a depression. Correct? कोई भी चीज आप बना रहे हो तो वहां पे से डिप्रेशन होगा. सो दोस डिप्रेशंस दे हैव अंडरस्टूड दैट दोस आर द एनपीसी. गॉट द पॉइंट? Okay, now another question is coming, that we expect that in the nucleus this NPC should be ऐसे systematically arranged, but what do we see here that एक जगह पे पूरे concentrate हो रखे हैं, right? Can anyone give me the reason why we see such kind of images? Any guesses? I mean, there is no definite answer, but I'm just asking this question to understand like your thinking, like how much you're able to understand and guess the answer. Yes. Yes, as usual, I have to take out the names. Akshaya, what is your thinking?
Maybe a particular part of the nucleus has been taken into consideration.
Okay.
And a lot of
Correct. And you can add also. You can add to this. Okay. Shruti, would you like to add to this?
I was thinking maybe it's an access issue, because I think the other parts are either being obstructed or like maybe part of endoplasmic reticulum शुरू हो रहा है क्या, वैसे something like that. There must be there's a reason why they are concentrated over there. The other parts may not probably need nuclear pores as much.
Okay, you have added 1 point. Okay, okay. So before I go to Tanvi, so I'll summarize. First thing is what Akshay said. Akshay, can you repeat? Sorry, I forgot.
I said that it may be a particular part of the nucleus.
Okay, so particular region has been taken. Okay, of course. And why this particular region has been taken? Because Shruti has said that because rest of the nucleus there might be ER coming, right? So there we won't expect much of the NPCs. Okay, and Shruti, sorry, Tanvi. Yes, Sunny, would you like to add?
Ma'am, maybe this particular region has a major part of depression, like that's why pores are there.
हाँ, see, depression is there, तभी तो वहां पे NPCs बहुत सारे हैं, but क्यों है? That is my question. So Akshay said that they have taken maybe this particular region, the screenshot, you know, in the electromicrograph. Shruti says that because rest of the nucleus there might be ER going on, है ना? जैसे अभी outer membranes हमने discuss किया. Okay, so anyways, this is correct. Of course, we do take the entire electron micrograph of the entire cell, Akshaya. The only thing is when we show, we obviously want to show the major part where the answer is there, right? So when they are studying about the NPC, they will show major regions of the NPC. So this is also correct. This is also correct, Shruti, because rest of the nucleus, they are, it is engaged with the ER. हम जब भी आपने structure देखा होगा, nucleus ऐसा रहता है. Rest of the structures we make it like this, right? ऐसे ER के continuous, ER से फिर यहाँ पे Golgi आता है, and then that's how we have studied the eukaryotic cell. So even this part is also correct. Another reason is that whenever the NPCs are over here, it means that that time they are busy. This is the moment at which the scientists have captured when the cell was actually exporting or importing the components. Are you getting the point? जब वो कुछ काम करने में busy था, so what they do is, like for example, I have to release so much of mRNA into the cytoplasm for the translation. So that time these NPCs will come together in one localized region because the signaling will be easy then. Correct? And then they start exporting out, or if you suppose have to import, so then that importing will be done. Okay, ये point समझ में आया? We'll see in further lectures that we have got यहाँ पे receptors, okay? To that your important proteins and exporting proteins. As I say, important यानी कि this is the protein which is responsible to import the components into the nucleus. ठीक है? Import मतलब क्या होता है? कोई चीज अंदर लाना, है ना? जैसे इंडिया में import होता है, तो यानी कि बाहर से import होता है. For example, अगारोज हम यूएस से इंपोर्ट करते हैं. एक्सपोर्ट मतलब इंडिया से बाहर गया. फॉर एग्जांपल मैंगोज हो गए, राइट? शुगर हो गया. देन अगारो सीवीड से बनता है, तो वो हम सीवीड एक्सपोर्ट करते हैं. So same way you have got the proteins called important and exportants. So from the name itself, it's very clear what kind of function they do. Okay, let's move on. Good. Okay, so underlying the inner nuclear membrane is a nuclear lamina, filamentous meshwork that provides the structural support to the nucleus. So this is like, you know, giving these back support so that the structure of the nucleus remains in 3D. It does not fall off. Okay, and it is composed of 60 to 80 kilodalton fibrous proteins called lamins, which is a class of intermediate filaments already mentioned. Now, these are the lamin proteins. This is how it looks like. So what do you see? It's your helical structures, right? So here, when you see in this structure, lamin A and lamin C, they come together like this. Okay, let us say that this is your lamin A. तो ये A हो गया, तो ये आपका C हो गया है. Okay, they come together and then they start forming the filament. Can you see the arrangement? So it's a dimer. This dimer again with the another one forms a tetramer, और ऐसी करते करते it forms the filament. This is the entire thing which is present below the inner membrane of your nucleus. Correct? Now, अगर ये आपका filamentous structure है which is made up of A and C, here like this, कुछ तो interaction होना चाहिए ना inner membrane और filaments के बीच में. So that is done by lamin B. Are you able to visualize this part? ये कैसा दिखता होगा?
Yes, ma'am.
Yeah, I'll just make it once again. The problem is in this notepad it's difficult for me to draw. Let us say that this is your nucleus. यहाँ पे आपका outer हो गया. Let us say that these are the NPCs. Okay. Then here, let us say that here is your lamin A and C का बना हुआ आपका filament. Okay. Now we need a connection between the inner nuclear membrane and the filaments. So ये ऐसे support करता है. Okay. So this is done by lamin B. Okay. So the association of lamins with the inner nuclear membrane is facilitated by the post-translation addition of lipid, in particular prenylation of C-terminal cysteine residues. So of course, see, lamins are ready. Now these lamins need to be aligned over there below the nuclear lamina, which is done with the help of the prenylation of the cysteine residues. ठीक है, यानी कि lipid में उसको anchor करना है, right? So that is done by the prenyl residues. So let us see what does this prenylation means. So prenylation is the type of prenylation shows affects RAS proteins and proteins of the nuclear envelope, which have cysteine residues followed by 3 other acids at the C terminal. The first step in the modification is addition of 15 carbon farnesyl group to the side chain of cysteine. This step is followed by proteolytic removal of the 3 C-terminal amino acid and methylation of the cysteine, which is now at the C terminus. ठीक है? So what it is saying is prenylation मतलब आपका cysteine residue में 15 carbon fatty acid आपने add किया farnesyl group, ठीक है? And then यहाँ पे जो 3 amino acids से those are being removed and it is being methylated. So this is how the entire protein gets inserted into the nuclear के inner membrane में. ठीक है? This is we have discussed before also, like N-myristylation कैसे होता है, palmitylation कैसे होता है. So this is like for the different your peripheral proteins. Okay. Now what you have to remember here is that for lamin, prenylation is happening. As you can see, it is also for the RAS proteins, it is also for the nuclear lamins. Okay, so here you need to remember this point. Okay, clear? Shall I go ahead?
Yes, ma'am.
Okay. Now, in addition, the lamins bind to the specific inner nuclear membrane proteins such as emerin and the lamin B receptor, directly connected to the cytoskeleton by protein complex called LINC complex that span the inner and outer nuclear membranes. Okay. Now don't get confused, it's just little complex. So right now I said that lamin A and C are connected to the inner nuclear by B. ठीक है, मैंने क्या बोला था, B से होता है. Okay, and all these connections are with the help of the prenylation. Sorry. So this is the chemistry behind it. Okay, lamin B is also a protein. Okay, so this prenyl group is helping in anchoring the lamin B, A, C in the nuclear envelope, inner nuclear envelope. Okay, along with this, we have got other proteins like emerin. Together they are forming a complex, LINC complex. Okay, with the cytoskeleton proteins. Now, what cytoskeleton proteins are talking about here? IF. And later these IF, okay, these lamins are again connected to the other skeleton proteins like actin. Okay, and lamins and the lamin associated proteins also bind to the chromatin. Now why? Later we'll discuss why and how it is binding to the chromatin, because in a nucleus each chromatin has got its territory. ऐसे रहेगा. Okay, when they are forming chromosomes, then this interactions breaks between the lamin and the chromatin, and then they form the chromosomes. And every time there is a specific location of each chromosome. ऐसा नहीं है कि अभी मैंने किया तो यहाँ पे 20th chromosome है और यहाँ पे आपका first chromosome है. So in that particular cell, this is the location where I can find the first chromosome, and this is the location where I can find the 20th chromosome. So in short, what I want to say is that every chromosomes or every part of the chromatin is having its own territory to be located. ठीक है, इतना समझा? Later we'll see how these chromatins are present in their territory. Clear? Will you be able to remember these proteins? Emerin, lamin B, LINC proteins. Okay, please do remember, and I'll try to share the video also as soon as possible. Okay, same the figure over here. Okay, so here, like now in the figure, will understand like what all I was asking you to remember. So we said that please do remember about the emerin, lamin B and LINC proteins, correct? Now let's see their arrangement. So what is this? This is your NPC, okay, nuclear pore complex. You can see that here these flagella or what tentacle-like proteins are present, right? Actually, these are helping in binding of the important and exporting proteins so that the respective components required by the nucleus can be imported and exported. Okay, we'll come to NPC in detail later. Now, also you can see that this is the outer membrane where you can see the ribosomes are also attached. What does it mean? Then it is in continuation with your ER also. Okay. Now, look at over here. So we have got the emerin proteins, right? So these emerin proteins are present in the inner membrane, inner nuclear membrane. Can you see it? Yeah. And it is making connections with the chromatin. These proteins, it is making connections with the chromatin as well as your lamins. Can you see the lamins जो पीछे की तरफ है? 1 second. This kind of zigzag, what do we see? Like this, like this. ये particular thing. Can you see? Those are your lamins proteins. And your emerin is there which is making connections with the chromatin. ये chromatin है जो बाद में इसके साथ connection तोड़ता है and becomes the chromosomes later. Okay, so what we have seen right now is that these LINC proteins or these complexes, they are actually making connections with the this LINC complex, this emerin, they're making connections with the lamins as well as the chromatin. Okay, did you get this point over here? Yeah, are you able to follow? So the inner nuclear membrane, they contain several integral proteins such as emerin, lamin B receptor. LBR is your lamin B receptor. Okay, it is binding to the lamins. And then we have got these sun proteins which interacts with the lamins. Now the sun proteins binds to the cache which is present in the outer, making up the LINC complex. Okay. Now again, which is connected to the nuclear lamina, and then this is connected to the cytoskeleton. So all this together is binding to your chromatin. So this association you have to understand. I'll just repeat once again. So you have got the proteins called emerin and LBR. Okay, LBR क्या हो गया आपका? Lamin B receptor. These two together binds your lamins. Okay. And lamins are again in connections with your chromatin. ये एक connection हो गया आपका. दूसरा क्या है? You have got the LINC complex. LINC complex में you have got two proteins. Sun, which interacts with the inner nuclear membrane. Then cache is there, which interacts with the outer nuclear membrane. Okay. Now these two together forms a LINC complex, which is again interacting with the nuclear lamins, which interacts with the cytoskeleton. Okay. In short, they all are helping in holding the lamins and the chromatin fiber. Yes, guys. Are you able to follow? Is this figure is clear to you? Figure clear है तो आपको हमेशा याद रहेगा कौन सा प्रोटीन किसको बाइंड कर रहा है. ठीक है? So yes, Akshay, Shruti, Tanvi, any questions in this topic?
No.
Okay. So I'll be stopping here now then.